All right, so ATP is made from ADP. Remember, all that we're really doing is putting that third phosphate on. And there's two ways we can do that. The first way is called substrate level phosphorylation. Sounds complicated, but phosphorylation is just a fancy word for put a phosphate on. Put a P on. That's all phosphorylation means, is put on a phosphate. And what's a substrate? It's the thing that the enzyme reacts with. We just did a whole chapter about enzymes and substrates. So literally, this has to do with enzymes. Well, substrate level phosphorylation, just like it sounds, is basically where an enzyme takes a phosphate from a substrate and sticks it on ADP to make ATP. That's all substrate level phosphorylation is. So if this was our enzyme, let's just say, and let's say this is our substrate, and our substrate has a phosphate on it, and over here we have ADP, and substrate level phosphorylation is basically where the enzyme helps to put the phosphate on. That's it. So it sounds complicated, substrate level phosphorylation, but what it is exactly what it sounds like. Just pull a phosphate off one thing and directly put it on to make ATP. So this is one way to make ATP from ADP. And it involves enzymes. The problem is it's not very efficient. Making ATP in this way, one at a time, you're not going to make a lot of ATP. But most organisms, including us, rely at least partially on this method of making ATP. As a matter of fact, per sugar, I've mentioned a couple of times, we make a total per sugar of 36 ATP. Only 4 out of the 36 are made this way. The other 32 are made by the other process. So obviously this is not very efficient if it's only getting us 4 ATP per sugar and we're relying on the other way of making most of our ATP. There are some bacteria that only do this though. Bacteria, remember, for one thing, don't have mitochondria. So most bacteria are using this way, literally taking phosphate off a substrate and making their ATP. They're only making a couple of ATP per glucose. As a matter of fact, the waste product of the bacteria contains a lot of energy. The waste product of most bacteria is ethanol, alcohol, which still has a lot of energy in it. Think about it. You can set it on fire. Alcohol burns. So bacteria are not extracting all the energy from sugar. They're not extracting it very efficiently. But they're bacteria. They're very small. They're not, it's not rocket science. They don't need a lot of energy. Okay, and again, this method of making ATP is anaerobic. It doesn't require any oxygen.
I got shot right now. The tranquilizer.
Good. Here's what it looks like. Enzyme, substrate, phosphate, stuck on to make ATP. Substrate level phosphorylation. It's making ATP directly from a substrate, 1 ATP at a time. All right, the other way of making ATP is called chemiosmotic phosphorylation. Chemiosmotic, what does that have to do with? Osmosis, which was what?
The diffusion of water.
Diffusion of water. Chemiosmotic phosphorylation has to do with movement across a membrane. It goes back to our chapter on transport. It's also called oxidative phosphorylation, not because it uses oxygen. Although oxygen is involved at the end, the name has to do with the fact that it uses redox reactions, which we talked about at the beginning of class. So oxidative or chemiosmotic phosphorylation uses redox reactions. This one occurs across a membrane. And what's going to happen in this case is we're not going to make ATP one at a time by pulling a phosphate off of a substrate. We're going to generate large amounts of ATP using the mitochondrial membrane. So you're going to have this mitochondria and this inner membrane, which we're going to talk more about later. And what's going to happen is you're going to have this flow going back and forth across this membrane of hydrogens and electrons. And what's going to happen is, over time, this is going to be sort of like turning little wheels, and you're going to generate large amounts of ATP. So you're not going to be building them one at a time by picking phosphates out. You're going to be doing it large scale, more like an assembly line, and this is going to be much more efficient. This is going to make a lot more ATP. So what's going to happen? Hydrogen is literally going to get pumped through to one side of the membrane, and then it's going to seep back in. And as it seeps back in, that's going to create a gradient for ATP synthesis. That word electrochemical gradient we also mentioned before, back in that same chapter on osmosis and diffusion. We only mentioned it very briefly. It had to do with a difference in charge on two sides of a membrane. I mentioned touching a hot stove and how it opened channels and caused positive ions to flow in. It may sound familiar. We only mentioned it briefly. But this chapter uses the same idea. Now we have a difference in charge, more hydrogens on one side than another, and that's going to cause this difference that when stuff flows through, it's going to generate ATP. And it'll make more sense later. This requires oxygen though. At the very, very end, oxygen is going to pick up those hydrogens. So you've got all these electrons flowing down a chain and hydrogens flowing in and out of the membrane. And at the very, very end, oxygen is going to pick up the hydrogens. And what do we get if we mix hydrogen and oxygen?
Water.
We are going to get water, which is non-toxic. So the final product in this type of phosphorylation is going to be water. And it's going to make a lot more sense later. This is just an introduction. All you really need to understand is that we can put a phosphate on ADP two ways. We can either use an enzyme, and literally the enzyme's going to help a phosphate get transferred, or we can use this method, which has to do with a bunch of stuff flowing across a membrane and generating large-scale amounts of ATP rather than one at a time from an enzyme. So that is the two ways we can make ATP. This is our overall reaction with glucose down here. C6H12O6, that's glucose, plus 6 oxygens. The whole point of the oxygens is that they are going to pick up the hydrogens at the end and make water. We end up with 6 carbon dioxides. This is our waste product. This is why the whole reason why we exhale carbon dioxide is because it's the waste product of this process. The whole reason we breathe oxygen is because oxygen is needed for this process. Without oxygen, we can only do the other way, substrate level, which means we can only make a couple of ATP per glucose. And then the rest of the energy is trapped. In the case of us, that's trapped in lactic acid, which is going to make your muscles burn, and you're not going to get enough energy from the glucose to run all of your cells. So we're going to generate, in our reaction, the whole thing through, we're going to be able to generate 36 ATP. And the goal of this chapter is for you to know where in the steps the ATP are generated. This is a very straightforward chapter. This is just showing if we released all the energy at once, you get an explosion. Instead, this is our electron transport chain. You've got stuff flowing through the membrane. Notice they don't even tell you what the stuff is. It's just electrons and hydrogens. And as that's flowing step by step, you're generating large amounts of ATP. So that's simplifying it. All right, so here's your basic steps. Your first step is called glycolysis. Glyco has to do with what?
Sugar.
Sugar, glucose, exactly. So glyco, glucose, and then does anybody remember what lysis means? If a cell lyses, what happens to it?
It's just literally breaks down or something.
Right. Remember in osmosis, if too much water went in, the cell would swell up and then it would lyse, right? Break. So lysis literally means to break. So glycolysis, all that happens is glucose breaks in half. That's glycolysis. Glucose splits in half. That's our first step in cell respiration. Our second step is called oxidation of pyruvate. Again, it has nothing to do with oxygen. Oxygen is not even involved. It has to do with oxidation. Pyruvate's going to lose either an electron or a hydrogen, and in this case, it's going to lose a hydrogen. Because that's what oxidation is. Remember, oxidation is loss. So pyruvate's going to lose a hydrogen. The third step is called the citric acid cycle. It's also called the Krebs cycle. Some of this may sound familiar from biology. The Krebs cycle, it's called after the person who discovered it. It's also called the citric acid cycle because the first thing that's made in the cycle is citric acid. You're not going to have to know all the steps in the cycle. You're not going to have to know all the steps in glycolysis. You really just need to know what goes in and what comes out. And then our last step is the electron transport chain. And again, just to summarize this, these 3 steps all combined only generate 4 ATP. The electron transport chain generates the other 32.
So most ATP is made in the electron.
Correct. So a majority of the ATP are made here. The thing is, these steps here build up everything that needs to go into the electron transport chain. It's sort of like going down a big water slide. In order to go down the slide, you've got to climb the stairs. And this is sort of like climbing the stairs. So all of these steps are necessary because they're going to build up a whole bunch of hydrogens for the electron transport chain, which is then going to generate all the ATP at one time. Well, you know, not at one time, not an explosion, but at one time in that sense. All right, and last thing, we have coenzymes that are involved in this. These are what are actually going to carry those hydrogens to the electron transport chain. Because that's what's most important are going to be in the electron transport chain are going to be the hydrogens. So what you're going to see is these 2 coenzymes, NAD and FAD, and what they're going to do is they're going to pick up hydrogens at all the other steps. At glycolysis, oxidation of pyruvate, and the Krebs cycle. They're going to basically go to all those places and they're going to collect hydrogens. When they do that, they're going to carry them. Here's NADH, FADH2. Is this reduced or oxidized? NADH. I got one reduced and one oxidized, but Kelly's right. It's reduced. NADH. Oxidation is loss, reduction is gain. NAD, no hydrogen, it gained a hydrogen. And FAD, the same thing. And these are going to carry the hydrogens. They're going to deliver them to the electron transport chain so that all the ATP can be generated. But they're going to pick up those hydrogens slowly. In glycolysis, they're going to pick up 2. In oxidation of pyruvate, I believe they pick up 2 more. In the citric acid cycle, they're going to pick up another, I don't know, 6, like 8 or 10. But the point is, they're going to then deliver those to the electron transport chain, and that hydrogen gradient is what's going to then turn those wheels to make all the ATP. So I'm going to stop